see the Mendeleev's experiment that he conducted. So first of all, Mendeleev made the homozygous tall plant. Basically, what is a homozygous or true breeding species means? Actually, like there are a trait that is known as the height. Height is basically so it's not a trait; it's a character. Basically, height is a character of plant stem. It can be tall, it can be short. Now the tall trait is dominant and short is recessive. So what Maldives actually do? He first crossed a pea plant so many times that after almost every generation, the pea plant finally produced tall seeds. And another another plant which he reproduces by self pollination again and again produce recessive, or we can say small plant again and again. So he known this as true tall plant, and he is known as true short breeding plant. Now, what he mainly do in first experiment, he first of all crossed it tall crossed with short. In the first generation, that this is the parent one generation, and this is the progeny, all were tall. He got four plants, and they were all tall. Now, for in the next generation, when Maldiv take this tall plants and cross them, he discovered that not all the plants that he got were tall, but out of all four plants, three were tall and one is short. So basically, that means that these plants which he crossed contain the short pyramid inside it, and they were known as the heterozygous tall plant. And Mendy at that time called them as called them as factor that basically determine the traits of the plant. So basically, for Mendy, it lists as the tall for TT is capital T, and for short is small t small t. So basically, what Mendy was tall, that is capital T, and for short take small t small t. So when he crossed, he got capital T and small t. He got capital T and small t, and when he again crossed, then he got capital T, and capital T small t for parent 2 generation, capital T small t. When he crossed them, then he got capital T, capital T small t, capital T small t, and small t small t. Basically, that small t represent the recessive trait as is shorter. So basically, what he actually do, he first of all he self pollinate the plant of tall plant again. That after every generation he got tall plant, he take that plant and again for do same thing for the short plant. Then he cross that tall plant with the short plant, and he named that factor as capital for tall and small t for short. Now when he crossed, he got capital T small t, he got capital small t. When he self crossed them, then he got capital T small t, capital T small t, and small t small t. So basically these both are heterozygous tall, and this is homozygous tall, and this is finally the recessive one. Basically, how he get find out that he again cross them and he again gets short plants. Basically, they are heterozygous tall, and when and even that's why same for the parent generation that they were also heterozygous. And if he crossed, then it again and again that we got capital T capital T, means a homozygous tall plant. Homozygous means if two same symbols are there, that's called known as homozygous. If there are capital in one trait, is called heterozygous. So basically, he got homozygous capital. For heterozygous, he got capital small t, and for recessive, he got small t small t. So this was the Meldy first experiment that he done. This data Meldy give the phenotype. What is the phenotype? Phenotype is basically the character appearance that is show. In the first parental generation, if F1 generation in the progeny, everybody was tall, so all tall. In the second, when you take the F2 generation, then they were three talls and one short plant. And when we talk about genotype basically the we can say the genotype means the genes that factor we represent. So basically these are genotype. The genotype in all he got, first of all, in all genotype that he got, that was because they were heterozygous, so capital small t, capital small t. That was 1:1 for heterozygous, because all were heterozygous tall. In the genotype second, when he got 1:2:1. That means one were homozygous tall, one were heterozygous tall, homozygous tall, heterozygous tall, and one were homozygous recessive. This is small tile, small short. So basically the genotype was 1:2:1 and the phenotype was 3:1.